Good morning, good afternoon, good evening, everybody. Welcome to the Ambassador Roundtable series hosted by Bioprotocol. Today, our webinar will be focused on protein-protein interaction techniques, an insight into modeling and experimental characterization of protein-protein interactions. We have the honor to have 3 speakers, and after each talk, participants will be able to ask any question through the chat box. So please feel free to type your question during the talk. We hope this talk will be very useful for students, postdoc, research scientists, PIs, and everybody in academia and industry that are specifically interested in using novel approaches to quantify protein-protein or lipid-protein interactions. It is my great pleasure to introduce Dr. Shayon Bhattacharya. Dr. Shayon Bhattacharya is a senior postdoctoral researcher in Professor Damien Thompson's molecular modeling group in the Department of Physics at the University of Limerick, Ireland, and the Science Foundation National Research Centre for Pharmaceuticals in Ireland. He holds a bachelor's degree in pharmaceutical sciences and a master's degree in pharmacoinformatics from the National Institute of Pharmaceutical Education and Research in India, and a doctorate degree in computational chemistry from UL. His current research involves developing and applying theoretical models and computer-assisted molecular design to provide rules for re-engineering biomolecular complexes in drug development. Please welcome Dr. Shayon Bhattacharya.
Thank you, Mariela, for that lovely introduction. So I'm just going to turn off my video so that it doesn't interfere with the bandwidth and everything, and then I'm going to share my screen if that's okay.
Please feel free to use the chat box to introduce your question at any time, and we will moderate it later on.
Can you see my PowerPoint presentation?
Yeah, we can.
Oh, perfect, Sean. Thank you. Can I start?
Yeah, please work away.
Yes, go.
Okay, thank you so much. Thanks very much, guys. Thank you, John, and thank you, Mariela, for organizing such a lovely webinar. And I'm really honored to have been invited to present my research for this webinar. So today I'm going to be talking on modeling protein-protein interactions for therapeutic target assessment and drug development. And as Mariela already said, that I'm a senior postdoc in SSPC and University of Limerick. And so I'm going to kick off with the very high-level definitions of protein-protein interactions and molecular dynamic simulations in general, because I imagine the audience is quite mixed here, so I don't want to go into too much of details, but, take you step by step and slowly unfold the events. And on top of that, also let you go through some of my works and how the techniques, the computational modeling techniques for quantifying or characterizing protein-protein interactions are involved or used in those works. So first of all, to start with, protein-protein interactions involves any physical contact, unintentional physical contact, or molecular recognition events that might be established between 2 or more proteins as a result of any biochemical event. So just going to show a laser pointer here. And so these have immense importance in playing important functional roles in cell-to-cell interactions, biological processes, and signal transductions. But not only limited to that, they also play an important role in defining the pathological pathways of several important diseases, and most of all, the neurodegenerative diseases or the amyloid diseases, which is going to be the main focus of my work here. So as you can see, when we talk about protein-protein interactions, you can visualize it as 2 protein subunits coming in close proximity to each other and forming a binding scaffold. And in between those, the PPI interface is formed here as highlighted. So what are the different types of protein-protein interactions? So they can be categorized based on three different types. So one is the compositions, other one is the lifetime, and the third is the affinity. So based on composition, we can further categorize protein-protein interactions as homo-oligomer and hetero-oligomer. So homo-oligomer being monomer chains stacked upon each other through different interaction types and same kind of monomer chains, whereas heterooligomers are different kinds of monomer chains interacting with each other and forming a heterooligomer. Based on the lifetime, the interaction could be transient or permanent. So transient interaction means they are reversible interactions. That is, two proteins can interact with each other transiently and can also fall back from each other. And they could be permanent interactions, that is, forming a complex over time that is carries out important functions. And taking on from there, there is also the third aspect, which will be affinity. And so based on that, the PPIs could be an obligate PPI or non-obligate PPI. An obligate PPI meaning that it is imperative for them to form those PPI complex in order to function properly, or for that matter, dysfunction, as I said, in diseases. And non-obligate PPIs, which are involved, basically, they could function on their own, the protein units, and also can function as a complex after they interact with each other and form the PPI complex. So the main focus of my talk today is going to be the most difficult PPIs to resolve experimentally, and why do we need modeling to do those? So the homo-oligomer formation, which is involved in self-assembly diseases or amyloid-forming diseases, the transient interactions that occur over time, and the non-obligate affinity of the PPIs. So this is going to be the focus of my talk. And so we need modeling, as I said, because of these particular PPI interaction types that are difficult to characterize experimentally, or for that matter, I mean, it's not certain. So we need the support of modeling in that regard. So for predicting PPIs by in silico modeling, there could be a number of different techniques. The first is the sequence-based predictions, which is the primary sequences of the proteins are used to derive certain rules based on similarity matching, that is conservation, co-evolution of the sequences. And then there are machine learning algorithms which could be used further to characterize the PPIs, which is hot topic nowadays. And then there could be computational proteomics approaches, which could lead more to a systems biology kind of prediction of the PPIs. The second is a structure-based prediction, and most common ones are the homology modeling and docking methods. The homology modeling is also based on the sequence similarity, and based on that, we predict the structure. And based on that further, the PPI complexes structure, the 3D structure of tertiary structures are predicted. And then docking is basically prediction of the binding interface, a static pose of two proteins binding together by using different kinds of algorithms. So one thing I must say that this is not exclusive. So there could be other different methods as well, but I have only highlighted the ones which are most prominent and important here. And the third one is the physics-based modeling, which is the molecular mechanics modeling of protein-protein interactions that is being that is the focus of this talk again, which is molecular dynamics simulations by using physics-based models. So jumping right into the molecular dynamics theory part, molecular dynamics simulations theory. So molecular dynamics is a method for determining the potential energy functions, which calculates the forces experienced by the atom relating to the positions of the other atoms in the protein. So you have these different atoms in the proteins, and based on that, you derive the forces and you give it a little push, which I'll show later through different slides. And then you derive the equations of motions from there. So as you can see that proteins have thousands of atoms, and they are pretty large in structure, although they could be small peptides as well. So only numerical solution is possible. So what happens is that consecutively for each of the atoms, the positions that is defined by the iterations through the time and the velocities are calculated over time. So a typical molecular dynamics simulation protocol involves, let's say you take a protein structure from the protein data bank, or this is a repository where the experimental structures of the proteins are deposited. But mind you, these are only static structures. So we need to derive important information from there. And for that, we need to run the molecular dynamics simulations. So if you want to see the binding of two proteins forming a PPI interface, then we would take the structures of those two proteins as a starting structure. And we would do some pre-processing by using several different, you could use several different softwares for that. And in our group, we use GROMACS, which is the most prominent molecular dynamics simulation running software. And then we prepare the system for simulation. So what we do is we basically put it in a box. We define a box around it with periodic boundary conditions, and we put water molecules in it, and we immerse the protein inside that water bath. And we include counter ions at physiological constants. Since it's a protein, so we have to take care of the fact that the biological aspects or the physiological concentrations and the buffer solutions are also taken into account. So basically, this is the sodium and chloride ions that are used as counter ions in order to neutralize the overall charge in the protein. And then we use certain simulation protocols like a thermostat or a barostat, pressure coupling, temperature coupling. So as you can see, the models are as good as the experiments. So we have to make sure that we maintain the in vitro or the in vivo conditions as closely as possible in order to accurately simulate the protein models. And then we run the simulations by using this high performance computing facility, which is the iCheck. And we do some post processing and we derive. So running the simulation is up till here, it's a standard protocol. And after that, the post processing and deriving of important properties and rules that we want to see through the course of the simulation is the most important part. It depends on what you want to see. And based on that, you kind of analyze the trajectories and derive graphs and data from there. So next, I want to give you a very high level step-by-step process of running this molecular dynamics simulation. So let's say you have defined certain time for which you want to run the simulation, basically. And this depends especially on the system you're simulating and also the fact that how relevant it is biologically. So if you want to see a process that happens like a folding process or something which would happen inside the body that would occur generally in the microsecond. So from anywhere between the microsecond to millisecond stage. So you define that timeline and then you divide or discretize the time into specific time steps. So just like this. So the frequency of integration is defined here. And for this particular case, you see every 1 femtosecond time step, we are integrating the motion of each of the atoms in the protein molecule. And the next thing is the calculation of forces. Now, mind you, this is the most expensive part of the simulation. So when you say the calculation of forces is generally done by something called force fields and also coupled with the water model, which I'll talk about briefly in the later slides, and also the equations of motion, which is basically Newton's law, the Newton's equation of motion of molecular mechanics. So we calculate the forces and we derive the connections between those atoms. So as I was saying, like these are defined by the molecular mechanics force field, which I'll talk about later. And the third aspect or the third step is basically you push the atoms a little bit. After defining the forces, you push the atoms with a little bit of velocity, not much, modestly, like a little bit, so that they get an initial kick and then they start moving. And finally, you iterate these steps over and over again and reiterate and integrate the Newton's laws of motion to obtain a trajectory of the atoms of the proteins and how they evolve over time. So as I was saying, the definition of the forces in molecular dynamics simulation is defined by the molecular mechanics force field combined with the water model. So, I showed you before that we immerse the protein structure inside a bath of water defined by the periodic boundary condition box, and that is being defined by specific water models. So the molecular mechanics force fields are nothing but a set of equations which defines the bond stretching, bond bending, torsion angles, electrostatic interactions, and van der Waals effect. So this is the most basic kind of force field definitions that you would expect or get. So the first three, in fact, the bond stretching, bond bending, and the torsion is generally regarded as bonded interactions. So that happens within the molecule itself. It's intra molecular thing between the atoms. So as you can see, if you regard the atoms as being balls and the bonds being formed between them as a spring, so if you stretch the spring, it extends, but it never breaks. So you can't break a bond during the course of a simulation. You can only stretch it, extend it as a spring, and then you can compress it. Similarly, between three atoms, you can bend the, change the angle by bending it during the course of the simulation. And then the torsion angles are basically the dihedrals proper and the improper dihedral angles that are defined by four different atoms being joined together in a plane. And for the non-bonded interactions, the electrostatic interaction deals with Coulomb's electrostatic charge-charge interactions. So, different atoms have got their partial charges, and based on that, they will either attract themselves or repel themselves from each other. And then you have the van der Waals interactions, which is basically depends on the radii of the atoms, how closely they are coming close to each other. I won't go into much details on these because I think overall, from the perspective of physics and chemistry, everyone knows that van der Waals interaction, how does it work, like the repulsion aspect and the attraction aspect of it. So yeah. And since we are dealing with atomistic simulations, I would only talk about explicit water models, that is the models that are physically defined, that are physically possible. They could be implicit water models as well that are defined implicitly based on certain properties. So unlike the protein force fields or the molecular mechanics force fields for defining the interactions within a protein molecule, for the explicit water models, these are defined only by the geometry and the non-bonded parameters. So you wouldn't have this bond stretching or bond bending. It's a static model here. So as you can see, there could be two different kinds of water models which are popular, although there could be a five-site water model as well, but that came up because of certain issues initially with the three-site water model, and then they tried to improve it, but that's obsolete now. So these two are the most prominent. As you can see, that the angle defined by this HOH is fixed here. It doesn't change over time. So does the H and the O bond distance and the bond length here. It doesn't act as a spring. So this is how we define a static model. It's defined only by its symmetry. But the non-bonded parameters are intact, as in like the electrostatics and the van der Waals between the two different water molecules are as defined by this protein forces. And then you have the four-site water model, which was basically developed because of an issue with the static charge distribution between the oxygen atom in here. So these are closely approximated with that of the experimental values. So experimental values of vaporization and all properties of physical water molecules. So these are very carefully parameterized. So with regards to the protein force fields, the most popular ones are charm, amber, OPLS, and bromose, with little difference in parameterization capabilities and the features in there. But all over, they are almost similar to what the set of equations represent here. And for the water modules, as I was saying, like we have T3P, T4P, and further during the course of their development, researchers came up with this other kinds of water models as well, T4P 2005, EW, which is the evolved distribution, and T4PD, which is the dispersion corrected water model, which greatly improves the protein water interactions. And it's mostly applicable for a different class of unfolded proteins called intrinsically disordered proteins, which I'll talk about later on. And so, as I was saying, like some proteins will require simulations for long time scales. Some proteins will require simulations for shorter time. If you want to see one particular feature within the process of PPI interactions, then you would require to simulate for a shorter time, whereas most of the physiologically relevant features are visible from anywhere between microsecond to millisecond scales. And we will need to have specialized physical models like combinations of protein force fields and water models for specific kinds of proteins like the unfolded proteins or the intrinsically resolved proteins that I'll talk about in this lecture. So let's jump right into the protein-protein interactions in peptide self-assembly. That is, finding drug targets for neurodegenerative diseases, as I was saying. So, neurodegeneration or neurodegenerative diseases are most common ones are the Alzheimer's and the Parkinson's disease, which destroys the structures of the brain over time, and it usually develops. It's an age-related disease, so it usually develops later in time, and it's difficult to catch these diseases as the symptoms develop quite later during the course of its development. So it's very difficult to assess at what point should the drugs be targeted. But in general, the process of the self-assembly of the proteins, the specialized class of proteins called the intrinsically disordered proteins, to be defined as a nucleated polymerization mechanism. And so initially, these intrinsically disordered protein monomers, that is a single chain of these proteins, they come together to different kinds of interactions depending on their vicinity. So they undergo transient interactions through primary nucleation to form this disordered oligomers, and then through conformational rearrangement, they give rise to structured oligomers, and then through seeded self-assembly to a long chain of protofibrils with the monomers or the intrinsically disordered proteins, which are no more intrinsically disordered. They are quite structured now. They're stacked upon each other at a specific order and structure. And then further through repeated polymerization, they may form this long chain of these mature fibrils, which through further elongation gives rise to these large plaques in the brain, causing Alzheimer's disease, or this neurofibrillary tangles, giving rise to the Parkinson's disease precipitation in the brain of patients. And they could be, so this is primary nucleation. There could be another mechanism by secondary nucleation where these monomers might get assembled on the surface of the preformed fibril or mature fibril and use it as a catalyst to form the structured oligomers, and then through further seeded self-assembly form the protofibrils and undergoes the whole process in this particular step. And then there could be fragmentation as well, where mature fibril could break into the disordered monomers, or for that matter, even into oligomers. So you can see it's quite a complex process. And therefore, you can imagine why drug targeting could be a difficult aspect for this neurodegenerative diseases. So the focus of my work was basically the initial pathway, where these intrinsically disordered proteins are able to form the nascent stages of the disordered oligomers. And these disordered oligomers, or for that matter, the structured oligomers even which are formed, which are basically beta sheet oligomers, are very difficult to characterize experimentally because they are transient. That is, they're elusive to experiments. As in the bulk experiments, when you try to mimic the process in vitro, the self-assembly process, then these oligomers are formed very, very transiently, and the time scale of the experiments are not, frequent enough to capture these oligomers, and therefore there is no, till date, there is no resolved structure of these oligomers for the proteins that are responsible for Alzheimer's and Parkinson's disease. But at a higher level, when they form the ordered structures, that is protofibrils and the fibrils, the structures are possible to derive basically. But at the initial stages, the nascent stages, so that's where we come in. That's where the modeling comes. And as you can see, the structured oligomers are found to be the most toxic species in the pathway. So just to give you a brief overview of how these proteins, the intrinsically disordered proteins, differ from a typical folded protein here. So this is an example of a simulation of the monomer of these proteins by itself. And as you can see, the folded proteins actually have their hydrophobic cores or the hydrophobic residues facing inside or inwards, which forms the cage. So the sticky patches are actually facing inside, and this lets the protein to be in a folded state. and it doesn't disrupt the structure of these proteins. Whereas for intrinsically disordered proteins, they transiently or randomly expose their hydrophobic residues outside, which is sticky patches outside, and therefore they are most amenable to interact with other monomers of these proteins, and the patches come together and stick with each other and form these oligomers, and in the process give rise to mature fibrils over time.
Sorry to interrupt you there, Shayon. I just want to let you know you have 5 minutes there on your time.
So 5 minutes left on my time?
Yes.
Oh, I'm already 20 minutes past. Okay. All right. Thanks, John. Thanks
No
for
problem.
that. I'll go through quickly, yeah. So these folded proteins are responsible for physiological function, whereas the IDPs are responsible for the neurodegeneration, as we can see. So the focus of this talk will basically be on the specific targets, the therapeutic targets. So there could be two different pathways. So one is the aggregation-prone pathway, and the other one is the aggregation-resistant pathway. So starting from the intrinsically disordered protein, they could form this partially folded alpha-helical monomers, which could give rise to beta-hairpin monomers, and then further for this beta-shaped oligomers, and then cross-beta-shaped fibril structures. And then in the aggregation impeding pathway, the resistant pathway, they could form this fully folded alpha-helical monomers. And then specifically for the protein responsible for Parkinson's disease, which is alpha-Synuclein, they could form this helical tetramers, which is also the native state of these proteins, and they are in dynamic equilibrium with that of the intrinsically disordered proteins. So these will be basically the targets that we'll focus on for this talk. So the first work where I want to show how to optimize the short-range protein-protein interactions in treatment of Alzheimer's and Parkinson's disease. So as I was talking about the force fields, for this work, we use 10 different force fields, combination of force fields and water models to define the folded, unfolded, and partially folded states of this protein. And these are the monomers of this protein system. And both these proteins, amyloid beta 42 and alpha-synuclein, have three different regions: N-terminus, C-terminus, and the central hydrophobic domain of the sticky region here. And as you can see from the contact mass, we find that the folded alpha-helical folded proteins have significant interactions, intra-protein interactions, short-range interactions between their termini and the central hydrophobic domain, which becomes sparing in case of the partially folded state, and the unfolded state completely lacks these interactions altogether. So we propose that optimizing the helical stability by re-engineering the terminal residues would be an efficient strategy to inhibit the pathogenic aggregation in Alzheimer's and Parkinson's disease. So next, we also wanted to look at how then the partially folded state actually promotes the aggregation of these proteins. So that could be an important drug target. And so we find that long-range interactions, inter-protein interactions, might be responsible for that. So we use a method called dynamic phosphorylation network analysis, which maps the intramolecular coupled atomic fluctuations from the molecular dynamics data, that is C-alpha, Pearson inner product phosphorylation coefficient. So these have got two components. One is the phosphorylation map, and the other one is the communication pathway. And this is defined by a set of equations. So as you can see, the Pearson's phosphorylation coefficient can be defined by the trace of the matrix and the submatrix that is formed. I'll go through that in a bit. So it's a 3x3 covariance matrix, which defines the fluctuations between the atoms in the residues, the atoms I and J within two different residues, basically the C-alpha atoms. So an overview of this method, as you can see, is we define different communities based on the cross-correlation, Pearson's phosphorylation coefficient. So this in this particular example, as you can see, over 0.5, the Pearson's correlation, when we have correlation coefficient over 0.5, then we can define three different communities within this structure. Mind you, these are all intra-protein interactions. And from there, we develop a consensus cross-correlation map. And we develop from there, we can map the probability density of the path length distribution. and the node degeneracy and between less centrality. I don't have time to explain this. I'm going to go through this quickly. And then finally, we can define the optimal and the suboptimal network paths from here, which will give us an idea about long-range interactions or communications between the specific residues or between specific atoms in a protein structure. So using this principle, using this theory, using this method, we were able to define specific interactions, long-range interactions of the F state, the folded state, the unfolded state, and the partially folded state, and we find that specific long-range interactions lasting between 10 to 20 angstroms were responsible for um, uh, between the terminus and the central hydrophobic domain were responsible in the partially folded states, which could further promote the aggregation of this partially folded state into amyloid structures. And these long-range interactions were lacking in the folded state and the unfolded state altogether. So we propose that targeting these long-range interactions, especially the termini of these proteins, could be an important strategy to uh, for the treatment of Alzheimer's and Parkinson's disease. So we also modeled as I was showing, the tetramers of alpha-Synuclein is in dynamic equilibrium with that of the monomers. So the shifting the population balance towards the tetramers, which is aggregation impeding, could be an important strategy to counteract Parkinson's disease. And for that, the tetramer structure again is very transient and it's difficult to characterize experimentally. So we designed different kinds of tetramer models to find the most stable one that we could use in order to define the shifting of the aggregation propensity towards the tetramers. And once it forms the tetramers, it will not further aggregate. So the idea behind it is to repopulate or increase the population of the tetramers of alpha-synuclein in Parkinson's disease. So as you can see by different models, the T4, that is our model, has a broken helical structure, and we optimize the hydrophobic packing within the monomers of these alpha helices. So since it's a tetramer, it has got 4 chains, and this inhibits aggregation in Parkinson's disease, and this is non-toxic in nature. So what we propose is that the designed Parkinson's disease associated mutant, the familial mutant tetramer models, have higher energy barriers in order to form those tetramers. Therefore, the mutants, that is the disease-causing mutants, do not form the tetramers, whereas the wild type is most amenable to form the tetramers because they have got a lower activation energy or energy barrier in that regard. So easily they can shift the population balance towards the tetramer. Following up on this work, we also wanted to find why the tetramer is ubiquitous, or why is it so populated in alpha helical form as opposed to other multimers or oligomers. So we again designed specific multimers, including the tetramers of the alpha-synuclein protein in Parkinson's disease, and we ran like 200 seconds, 200 nanoseconds molecular dynamics, and we find that the tetramer is the most stable amongst all oligomers. And it also shows least activation energy amongst all oligomers, as you will see in the next slide here, where the activation energy for the formation of the tetramer is the least as opposed to all other oligomers that are formed during the course of the simulations. And with regards to the PPIs between the monomers of the tetramer, we also computed the pairwise interaction energies. And as you can see, there's a subtle balance in the formation of the tetramer between the protein-protein interaction and the protein-water interaction. So stronger protein-protein interaction is found in case of a tetramer than the protein-water interactions that would favor the formation of the tetramer as opposed to the other multimers. So this is a very important finding, because leading on from here, we were further able to define the population balance of the formation of the tetramer on top of the membrane, where the protein-protein interactions between the monomers of the tetramer against the protein and membrane interactions of the alpha-synuclein with that of the membrane. And we find that depending on the membrane environment, they were able to form different kinds of morphologies of the tetramer. So we designed 3 different models of the tetramers and put them on top of the membrane, the biological membrane. So depending on the membrane composition, we find that the population of the broken or an extended tetramer were different. And this could be an important finding because if we could experimentally design liposomes, which mimics the membrane composition, and we can capture the tetramer in their either extended or broken form, that could repopulate the tetramer, and that could be an important strategy for the treatment of Parkinson's disease. So moving on from there, I just want to highlight a few more works here, if we have, in the interest of time. How far am I, John?
So there hasn't been any questions on the chat yet, so we just said we'd leave you go a little bit longer if you want. But if you could just wrap up within the next minute or two, that would be great.
Okay, that's great.
If that's okay. No, there's no problem. There's no problem. And I just look while you're there. If anyone does have any questions, I'm going to put Shayon's email into the chat. So if you do have anything that you want to ask him, please, you can just copy the email and ask him questions by email. Alternatively, he might be able to respond as well because he's a co-host on the webinar. Thank you.
Perfect. Sounds great. Thank you.
Thank you so much.
Thank you, Marian. Just very quickly, I'll try to wrap this up in a couple of minutes. So the next work that I'm going to talk about is the collaboration of SSPC with that of experiments, basically the experimentalists, the collaborators that we have got. So as I was saying, the models are as good as experiments or the in vitro and the in vivo experiments that have been carried out. So I have put together two different works here because they're based on the same kind of philosophy, using a surface to delineate the pathway of the aggregation of these proteins. So the first work is actually both of these works are in collaboration with Peter and this group at EMPA in Switzerland. So on the work on the left, we show the so Peter performed some atomic force microscopy experiments on the graphene, so using the graphene water interface. And the reason for using a platform like this is because we can study. So these proteins aggregate very fast if you perform this experiment in bulk water. Therefore, we need to use some kind of a platform in order to characterize the events that operate, so that we don't miss out on any of the assemblies or the aggregates that are formed over time. So that's why the graphene was used as a platform. And through AFM experiments, we found that the population of the amyloid beta 42, which is an alloform in the formation of the Alzheimer's disease, which is the most toxic one, and so it's 42 residue long, is formed at a so there's a crisscross of assemblies that are formed on top of the graphene, which was not evident for the less toxic amyloid beta 40 alloform of these proteins, and it was formed in more unidirectional manner. So we wanted to figure out through modeling why this happens. So first, we computed the most probable orientation of amyloid beta oligomer on the surface of the graphene water interface, and we find that orientation 3 is most amenable based on the conformational energies, or the binding energies for that matter, with that of the graphene. So we find that orientation 3 is most amenable for both these amyloid beta 40 and 42, which means that the aggregation will take place parallel to the surface of the graphene. And then we compared our model heights with that of the experimental model height that Peter derived from the AFM experiment. And based on the hydrogen bonds, which is the PPI hydrogen bond interaction between the monomers of this protein, we define that the three ends of these oligomers have the propensity to form different occupancies of the hydrogen bonds. As the percentage of formation of the hydrogen bonds differed between the two ends of these oligomers, which is an important finding because it points towards a bidirectional growth of amyloid beta 42, whereas a unidirectional growth of amyloid beta 40. So this is one of the morphological differences or signatures between amyloid beta 40 and amyloid beta 42 that was not found, and this can be an important feature that would highlight the more toxic nature of amyloid beta 42. In the second work, we performed the experiments on alpha-Synuclein and for that matter modeling as well on top of the gold water interface. So we simulated the different constructs of the monomers and oligomers here, And in close proximity, in close collaboration with that of the experiments, the AFM experiments, which is also conducted by Peter's group, we find that for trimers, especially not for the other multimers, for trimers, there is so as we go on increasing. So this modeling was performed in presence of metal ions such as copper, because copper is known to um, uh, increase the toxicity in the formation of the Parkinson's disease, and in the brains of the Parkinson's disease patients, there was a high concentration of copper found. So therefore, we wanted to find the dose-dependent effect of copper on the alpha-synuclein aggregation. And for specifically trimer, we find that as we go on increasing the concentration of the copper, the trimer becomes less and less stable, which was not evident for the other oligomeric constructs where they become more and more stable here. So this was a typical case. And so we further went on and found that at low copper concentration, the trimer is actually um, uh, forming a traditional um, aggregation um, aggregating oligomer, as in like it's aggregating parallel to that of the surface of the gold. It has the propensity to aggregate in this direction, whereas at higher concentration of copper, it forms a completely different morphology, which does not have the propensity of aggregating on the surface of the gold. And we correlated that closely with the AFM experiments, which showed that a typical alpha-synuclein protofibril is formed at low concentration of copper, but as you go on increasing the concentration of copper, the morphology of these oligomers completely change, and they form this typical annular alpha-synuclein protofibrils, which was not observed previously. And this could be an important drug target, basically, for Parkinson's disease.
Shayon, we're
So,
going to have to leave it there, I'm afraid, if that's okay with
okay.
you.
Yeah, absolutely fine. So I'm just
Super.
gonna pull up the last few slides. So it's just a couple of slides. So these are all our collaborators, and thank you again for your time. And I'm sorry that I ran over time. And you can visit my YouTube channel, and please like, share, and subscribe. So this has got molecular modeling playlist. And thank you once again.
Thanks to
Thank you
you.
very much. This is a very interesting talk. But like, so you basically, if anyone has any questions, just please get in touch via email in the chat. So Mariella now is just going to take over and introduce the next speaker. Thank you so much, Shayon. Really good presentation. Very enjoyable. I'll be in touch with you soon with some questions for you.
Thanks
Thanks,
very much,
John.
John. I look forward to it. Thank you.
Thanks, John. So our second speaker is Vinay Kumar ldikuda. Vinay Kumar ldikuda did his bachelor's in pharmacy from Osmania University in India. He later pursued his master's in pharmaceutical science at Idaho State University in the United States, where he studied metal oxide nanoparticle toxicity in Dr. James Lay laboratory. During his PhD at Virginia Commonwealth University, he studied ligand binding in HCN channels using electrophysiology and fluorescence-based methods. Currently, in the lab of Dr. Bharat Shandat at Washington University in St. Louis, he's doing his postdoctoral, which aims to understand the role of lipids playing in ligand binding to ion channels using single molecule spectroscopic methods. Please, Dr. Kumar ldikuda, welcome.
Thank you. Let me share my screen. You can see my screen, right?
Yep.
All right. Yeah, thank you. Thank you, Mariella, for the nice introduction. And I'd like to thank John and Mariella for sending me an invite. Let me just skip the video so it's easier. Yeah. So the title for my talk today is Single Molecule Approaches to Study Protein-Ligand and Protein-Protein Interactions. The crux of the talk will focus on protein-ligand interaction because that is what we have been characterizing in our lab. But then I will very briefly show some of the work that we are right now doing to study protein-protein interactions as well. Okay. Oh. Okay. So this is the overview of my talk. I will briefly introduce two HCN channels, and then we will talk about ensemble versus single molecule measurements. And then I'll talk about single molecule ligand binding using zero mode waveguides, and then protein-protein interactions using zero mode waveguides. Finally, summary and conclusions. So in our lab, we work on HCN channels. These are hyperpolarization-activated cyclic nucleotide-gated ion channels. These are membrane proteins with a large membrane spanning region and a small cytosolic domain. As I mentioned, these are ion channels, so they open upon hyperpolarization. And once these channels open, they allow cations like sodium, potassium to enter into the cell, and thus depolarizing the cell. These channels also have a soluble cytosolic domain to which cyclic nucleotides like cyclic AMP bind, and they regulate the channel activity. So in a normal case, when cyclic AMP is not bound, the channels open, and you can measure these using electrophysiological methods like patch clamp, and you will see an inward current. But when you add cyclic AMP to the solution, cyclic AMP binds to the cyclic nucleotide binding domain, and then this will make the channel opening faster and more complete. So the basic question that we in our lab are interested in is, how does cyclic AMP binding regulate the channel activity? So as Feynman famously said, it is very easy to answer many of these questions if you just look at this thing. So in a bulk measurement, which these experiments have been done before, is you express these ion channels in an oocyte, and then you can pull out a piece of this membrane from the oocyte, and then you have all the channels in this piece of membrane. You then perfuse a fluorescently labeled cyclic AMP, and then you can measure both fluorescence and current recordings at the same time. In this one, what you can see is the fluorescence coming from the fluorescent cyclic AMP binding to the patch, and currents from a patch clamp recording. As you can see, when the channel is open, you see an increase in the fluorescence. So as the channel opens, the more and more ligand binds to the channel. So this is how you can measure a bulk ligand binding to the channels. But the problem is that what you're seeing is an average result. There could be information that is hidden inside this average information, which is subpopulations, kinetic states, and transition states, which all this information bulk binding cannot answer. So there is a need to measure these ligand binding at single molecule level. So this is our approach, is to watch direct binding of fluorescently labeled ligands to HCN channels using single molecule fluorescence. But the real problem is that diffraction limit of light. So to be able to individually resolve two molecules, they should be at least separated by a distance of diffraction limit of light, which is roughly half the wavelength of the incident light. So in a normal epifluorescence microscope, if you have fluorescently labeled molecules, you would excite bulk solution, and then you would have a lot of noise. So what most single molecule experimentalists do is use a TIRF mode, which is total internal reflection fluorescence microscopy. In this case, the light is incident at an angle which is greater than a critical angle. This will allow a total internal reflection. This will generate an evanescent fluorescent field above the plane, and this evanescent field decays exponentially with distance. So what in essence happens is you will have only an excitation field of about 100 nanometers above the surface. So that bulk of the fluorescent molecules are not excited, but the ones that are close to the surface are excited. So in this way, you can go up to 10 nanomolar. For example, you can take a cover slip and tether your receptor or ion channel in our case to the cover slip, and then perfuse freely diffusing fluorescent ligand. You can then incident your laser on this channel, and then there is a decaying evanescent field. The molecules that are bound to the protein will fluoresce, and then you can then measure this in terms of either binding or unbinding. But this can only be done up to 10 nanomolar fluorescent ligand. So you will be able to differentiate between a signal versus background. But once your protein, your fluorescent label concentration exceeds certain amount, for example, in the micromolar range, you will not be able to discern the signal versus noise. So this becomes difficult to measure activity at micromolar concentrations. And also most biologically relevant interactions occur in the micromolar and the millimolar range. This is a histogram of affinities of about 30,000 enzymes that is taken from a Brenda database. And here you can see that most of the enzymes have affinity that is in the range of micromolar or millimolar. So we need a method that can measure single molecule activity, but at micromolar and millimolar range concentrations. To do that, what we do in our lab is we use zero mode waveguides. Zero mode waveguides are nanophotonic devices. These are just you have a silica substrate on which there is an aluminium coating, about nanometer thickness aluminium coating. And then you have these holes that are etched onto the aluminium, which is about 120 to 200 nanometers in size. Now you can have your protein, which is sitting in each of these wells, and this will having an aluminium layer around it will further decrease the excitation volume. And in this way, you can go to micromolar or millimolar concentrations in certain cases. So what happens is you do the same experiment, but instead of just having a cover slip, you have an aluminium coating that is surrounded by that the protein is surrounded by. This will then allow you to image in micromolar ligand concentrations. So basically you're going from a femtoliter observation volume to a zeptoliter observation volume. So using a zero mode waveguide, you can tap into millimolar and micromolar ranges. If you can do a FRET experiment on top of this, then you can further go into tens of millimolar concentrations along with the ZMW. So we get our ZMWs made from Oak Ridge National Laboratory, which is where one of our lab mates goes and makes these ZMWs for us. Another big advantage of using a ZMW is that you can do a high throughput analysis. So this is a bright field image of a ZMW, and you can see that there are about 1,600 wells in this single field of view. So what you can do is you can label your protein in this ZMW, and you can simultaneously measure activity from many number of molecules, as you can see here. Maybe I need to. So what you're seeing here is ligand binding to monomeric cyclic nucleotide binding domain of HCN channel. I have highlighted some molecules in yellow here, showing activity, binding activity, and you can measure this binding activity. So what I have shown you before is a monomeric cyclic nucleotide binding domain binding to cyclic AMP. But as I mentioned in my first slide, these ion channels are tetrameric ion channels. So what we then did was to put the tetrameric channel in the ZMW. We first coated the ZMW with a PEG layer, biotinylated PEG, and then next a coating of streptavidin, and then a biotinylated GFP trap. This is nothing but a GFP antibody. And then we have our protein, which also expresses GFP on the N-terminus. So we could pull down the protein using this GFP nanobody and then perfuse cyclic AMP, fluorescently labeled cyclic AMP. So what we do first is identify which of these wells have the protein by the GFP molecule. We excite it with a 488 nanometer laser, so we can identify which wells have the protein, and then we can measure the binding activity from the corresponding wells. What this will give you is a state occupancy diagram where you can see some of the times the protein is bound to just one molecule, but then you can also have up to 4 ligands bound to the channel. And as the concentration increases, you will see more and more of the protein binding to 3 and 4 cyclic AMP molecules. So basically what you do is you get your data and then you idealize this data. This information, what you get from this is whether you're looking at a signal or a noise, and how many states are involved in this, and how the transition between these states is happening. And then you can uncover the dynamics such as bound state versus unbound state. So what other information you can get from this is you can also get a state occupancy histogram, as I mentioned before. For example, in this case, when you have 100 nanomolar cyclic AMP, most of the molecules are in about 60% of the channels are in unbound state. And the rest of the 40% is in either 1 bound, 2 bound, 3 bound, or 4 bound state. But as you increase the concentration to 1000 nanomolar cyclic AMP, you can see more molecules in the 3 and 4 and 5 bound states. And also, as I have mentioned before, this is a very high throughput process. So you can just look at 83 molecules for each of them for 10 minutes, and then you can have about 13 to 14 hours of binding data, and this will give you a very robust amount of information that you can later quantify. What you can also measure is the amount of time the ligand spends in the binding site. From this information, you can also find out if there are more than one bound state, or how if there is more than one bound state. For example, in our case, we always were able to fit our data better with a bi-exponential, suggesting that we have 2 bound states. Once the ligand comes and binds, and then there is a conformational change that leads to a longer bound state. So this is all very useful information that you can get from the data just by doing single molecule ligand binding experiments. So because the talk for today's, the focus for today's talk is on protein-protein interactions, I just wanted to briefly show that we can also extend the same studies to protein-protein interactions as well. One of the things that we are doing in our lab is to look at an accessory subunit of HCN channel, which is called TRP8B. This TRP8B binds to the HCN channel, and shown in yellow is the domain that binds to the HCN channel, which is about 80 amino acids. So we purified this peptide, and then we wanted to look at what stoichiometry it binds to the HCN channel, and what is the true affinity of the binding interaction, and whether or not any conformational changes are happening. We just started to do these experiments, so we don't have too much data, but what I can show you is definitely you can also study not just protein ligand, but protein-protein interactions using ZMWs, where you can see this is very low amounts of TRP8B, but we were able to see the TRP8B binding to the HCN channel in more than one state. We also recently did some experiments using GFP labeled G protein coupled receptors. This is data collected from one of my colleagues, Suzuwan Chaudhary, and we were able to even look at binding of a G protein to a G protein coupled receptor. Again, a caveat of all of these is you need to have a labeled protein. So we were able to label the G protein with a SNAP Alexa 546 and looked at the binding of a G protein towards G protein coupled receptor. So in summary, I think I was able to convince you that single molecule measurements will give us valuable information about subpopulation state sequences, which otherwise are impossible to get in an ensemble measurement. And using TIRF microscopy in conjunction with zero mode waveguides will allow us to do single molecule imaging in micromolar range. And if you can use FRET, you can even tap into millimolar range concentrations. And ZMWs are very useful to measure both protein ligand and also protein-protein interactions as well. I would like to thank my lab mates. This is a picture of our lab, and would like to thank my PI, Barun Chanda, for letting me do all these experiments and explore all this fancy technology. Suzuwan Roy Chaudhary, he gave me some of his GPCR data just to show the protein-protein interactions. and then all other lab mates, our collaborator Randy Goldsmith at UW-Madison and Oak Ridge National Laboratories for allowing us to make ZMWs there. Thank you, and if you have any questions, I'll take them now. I hope I didn't rush through.
So I think we have a question. Is the
Sure.
HTS method also useful to characterize Shayon peptide interactions, or do they always need to form a stable complex? Thank you, Vinay, for a lovely talk. This is a question from
Thank you.
Shayon, our previous speaker.
Could you repeat the question? Sorry.
Is the HTS CMW method also useful to characterize transient peptide interactions, or do they always need to form a stable complex?
You can even measure transient complexes, but at least the amount of time it binds to the peptide should be greater than your frame rate, which is 100 milliseconds in our case.
We have another question. How long does it take to develop a functional assay for a new interaction?
If you have a TIRF microscope and you have access to ZMWs, it would not take long. But I think those two are the limiting factors.
I see. Maybe I missed it, but how do you quantify the yield of labeling?
Yeah, so we measure the we use a cysteine label. So we use a maleimide dye to label the protein, and then we measure the protein concentration, and then after labeling we measure the protein concentration as well as the dye concentration, and there is a way to get the labeling efficiency from there. Yeah. But our ion channels are GFP tagged, so we purify them with the GFP, so there is no question of not having a label on it.
I see. Okay. Thank you. Is there any other question? Please feel free to ask. So thank you so much, Vinay, for your talk. We really appreciate it. And feel free to contact him through email. Please don't hesitate to contact him. So our last speaker is Dr. Hannah Kratzat. Hannah Kratzat is a biochemist with experience in structural biology, protein biochemistry, and molecular biology. She obtained her bachelor and master's degree in biochemistry at the Ernst Morris Arndt University in Greifswald. During her PhD at the Ludwig Maximilian University in Munich, she worked on protein translation and protein quality control using cryo-electron microscopy. Today, she works as a field application specialist at Nanotemper Technologies, covering the UK, Ireland, and Benelux regions. During her talk, she will cover the basics of MST, which is microscale thermophoresis, a powerful method which allows you to directly measure the strength of molecular interactions in solutions, along with latest applications examples in the field. Please welcome Hannah for her talk.
Thank you so much. I hope you can hear me fine, and
Yes.
you can see my slides.
Yes, we can hear you.
Thank you. first I would like to thank Mariella and John for organizing this very nice webinar and giving me the chance to speak. And it's my pleasure to introduce to you the monolith and show you how you can use this instrument to characterize your challenging interactions. But before we dive into the technology and application examples, I would like to give you a brief overview of the history of nanotemper. So Nanotemper Technologies was founded in 2008 based on the scientific work of the co-founders and CEOs of our company, Stefan Du and Aske. And just 2 years later, then the first serial instrument was released, which is called the monolith. And this is basically the first version of it. And then to meet the needs of our growing industry customer base for higher throughput, we developed an automated monolith instrument, which was released in 2014. And at the same time, we were also able to release our second patented technology platform, which is the Prometheus instrument, and again, quickly added an automated solution for high throughput measurements here. This line of products actually focus on the protein stability measurements. In 2019, we then added the Dianthus to our portfolio, which is a device dedicated to high throughput screening of interactions. And in 2020, the Prometheus Panta was launched, which in addition to protein stability measurements also allows you to analyze aggregates and sizing of your sample. And last but not least, in 2020, also the new monolith was released, which is the star of today's presentation. And I'm happy to talk more about this now. So as I said, the monolith is a biophysical tool to characterize interaction. So what type of molecules can we analyze with the monolith? As you can see here, you can work with many different types of molecules ranging from proteins like antibodies, membrane proteins, and enzymes to small molecules, nucleic acid molecules, but also large particles as vesicle cells or virus particles. And as today's topic is protein-protein interactions, and you know how protein-protein interactions have a pivotal role in all life processes, it's really important to understand how these interactions work, and it's crucial to have this understanding to then be able to develop drugs for a lot of disease. And this does not only include understanding how one protein interacts with another protein, but also, for example, how protein-protein interactions can be modulated, or protein complexes form an interface that is bound by a third component, so looking at ternary complexes. And at Nanotemper, we develop biophysical tools to characterize the stability of proteins and interactions. And so we know how difficult it can be to characterize certain targets, and targets like membrane proteins, for example, were for a long time considered to be even undruggable because they're so difficult to handle. And some proteins are still very, very difficult to characterize. But our goal here is to find new tools and methods to overcome these challenges. And here are some reasons why it can be difficult to biophysically characterize these targets. So some are just very difficult to immobilize, or you have low concentrations or low sample volume of these. And I think everyone who worked with membrane proteins knows how difficult it can be to preserve their native conformation or get high amounts of these proteins from protein expression. And the monolith offers one solution here. So you can actually analyze these proteins in solution. Let's say if you have a membrane protein in nanodisk or liposome, and you can actually use any buffer with detergents or TMSO you like. So you can also use quite low amounts of samples for these kind of measurements. And similarly, if we look at intrinsically disordered proteins, because they don't fold into a homogeneous three-dimensional structure, they're prone to aggregation, and especially at high protein concentrations. So monolith resolves these limitations by allowing for low protein concentrations in solution measurements, and it even allows to differentiate between aggregation and binding. Another interesting target that we could look at is proteolysis targeting chimeras, in short PROTACs. And here the monolith also allows you to efficiently evaluate ternary complex formation. And during my talk, I will now first explain you the technology that is used to analyze these interactions, and then later show you examples where this method was used. So the technology is called microscale thermophoresis, short MST. And MST measures the strength of an interaction by measuring the changes in fluorescence. So the change is a result of an infrared laser-induced temperature change, and the fluorescence that is detected comes from a fluorophore that is attached to the molecule of interest. And there are actually two components that contribute to the fluorescence change that we measure then. So one is called thermophoresis, and this is the directed movement of molecules within a temperature gradient that then changes the concentration of the fluorescent molecule. And the extent of the concentration change here really depends on the molecule's overall properties like size, charge, and conformation. And so that's why when we have a ligand binding, it can have effect on how the molecule moves as soon as we apply a temperature change. The other component of the MST signal is called temperature-related intensity change. And this just means that even if the molecule would not move away from the heat, but just stay in the same position, the fluorescence could change just because the fluorescence is temperature dependent. So we have the following nomenclature here. So we have the target that is always fluorescently labeled, and then we have a ligand which of course cannot be fluorescently labeled. And we would then measure the effect that the ligand binding has on the fluorescence of the target. So there are two ways of, or two mechanisms how the ligand can actually change the fluorescence of the fluorophore that we have attached to the molecule. So on one hand, we have the case where the fluorophore is attached in close proximity to the ligand binding site. And therefore, once the ligand is bound, this chemical environment around the fluorophore is changed, and that's why we see an effect. On the other hand, we can also have the case where the fluorophore is in a distant position from the ligand binding site. But here also the chemical environment can be changed by a ligand-induced conformational change, which can be actually quite small, which then still changes the chemical environment of this fluorophore and therefore leads to a change in behavior. So how would a MST experiment look like? So we basically just monitor the fluorescence over time, and we look at different states, the unbound state and the bound state. And we can see that as soon as we turn the IR laser on, we have a reduction of fluorescence, and that's because the fluorescence changes because we have a temperature change, but also because the molecules most of the time move away from the heat that is applied. And so the way the molecule behaves here is different for the bound and the unbound state, which in the end gives us this MST signal amplitude. And this is now how a real example, like a real experiment would look like. So first we look at the MST traces for the unbound state, where we measure the fluorescence before we turn the IR laser on, and then after the IR laser is turned on. And then we would basically get a binding curve by looking at different concentrations of the ligand. So we do a titration series of the ligand, and for each one we then analyze the MST traces, and then we plot the F1 over the F0 fluorescence, and we can thereby then derive a binding curve from which we can determine the KD and the binding affinity. And to give you some examples on how this could look like, I want to show you some application examples now. So I want to start with an example where MST was used for hit validation. So the molecule we look at here is an epigenetic regulator called ATAD, and it has been linked to the progress of various cancer types. And it has always been a very difficult target just because there are so many isoforms, so you really need a very specific ligand here. And what the authors did is that they ran a DNA-encoded library screen, and with this discovered the ligand BAE850, which is a selective and potent inhibitor that actually induces dimerization of this molecule and thereby prevents interaction of the molecule with acetylated histones as well as chromatin. And this was like a very interesting study because they first ran the screen, and then from the hits that they got, used different biophysical methods to confirm that this binding is real, and also to better understand the binding. And here they found an affinity of 85 nanomolar of the BAE850 to this protein. The next example here shows the binding of a misfolding-prone antibody to a chaperone protein. And so this antibody is actually really related to some diseases in terms of that certain mutations lead to aggregation or fibril formation of this antibody domain. And there is a chaperone that actually binds this antibody domain. And so here the authors basically measured the affinity of the chaperone to the antibody domain, the wild-type antibody domain, and the mutant antibody domain, and found that these mutations actually cause an increase of affinity of this chaperone to the antibody domain. Next, this example is a membrane protein. It's a transporter that is responsible for nitrogen assimilation in plants. And this transporter was expressed as a GFP fusion and purified and characterized in the presence of the detergent DDM. And here the binding of nitrate is evaluated for different mutants of the transporter. And that's quite interesting because this transporter is expressed in conditions where we have quite a high nitrate level outside the cells, so in the soil. And to basically, when the nitrate levels drop, then this threonine here, which they mutated, is phosphorylated and leads to the transporter to be able to have a higher nitrate uptake. So the first question was, when we change the threonine, does the affinity to nitrate change? And they actually found that it doesn't change, so it stays the same. So this is actually a different mechanism, which they could later on with the structure determine that the flexibility of this transporter changes by phosphorylation of this, which leads to the higher uptakes of nitrate. But they also then were able to find another residue, which is this histidine 356, which is responsible for nitrate binding, and they could confirm this with this MST experiment here, where you see no binding. The next example is a very big protein complex, which is over 800 kilodaltons, and it forms functional 3D structure composed of 3 independent dimers. And in humans, this elongator complex appears primarily to affect the development and maintenance of nervous systems, as some individuals suffering from certain neurodegenerative diseases carry mutations or variants of one of the 6 human elongator genes. And it's quite difficult to purify such a large complex. And so for the authors of this studies, it was very important to have a method where they could use low sample volume and still get an affinity. And here they basically test different tRNAs and determine the affinity of these tRNAs to this complex. In this next example, I want to show a pH-dependent binding, which is quite interesting. So binding of IgG, which is shown in blue here, to the Fc receptor, which is shown in green, actually prevents degradation of Ig in endosomes. So it's quite important to understand the retention time of drugs in blood when you want to develop a therapeutic. And so this is quite a nice example how this works. So IgG actually at pH 7, as you can see from the binding curves up here, has not a very high affinity to the Fc receptor at pH 7. But as soon as the pH drops, you get a very high affinity, and that's the case in the endosome. And it's also then how the IgG can be released after getting back to the serum where we have a higher pH. Sorry. This example shows an interaction of two proteins. So here we have the middle luminal domain MLD of the Niemann-Pick C1 protein, which is involved in cholesterol transfer to another protein called Niemann-Pick C2 protein. And actually, again, here we have loss of function mutations in this protein, which actually lead to a lysosomal storage disease. And these can lead to death because of accumulation of cholesterol in the body. And the group selected several of these mutations of the NPC1 and then measured the interaction to NPC2 with MST. And you can see the binding curves here, and depending on the mutation that is present, these can vary. So that was quite important to understand how this cholesterol transfer happens. This is the last example that I want to show for MST. So here the effects of membrane fluidity on GPCR function is analyzed, and the human serotonin receptor was for these studies removed from its biological membrane and constituted with a GPCR nanodisk. And for creating this nanodisk, actually different phospholipids were used, and then the nanodisk itself was actually fluorescently labeled to then analyze the binding of serotonin to this receptor. So you can see that for the different nanodisks, the affinity of serotonin to the receptor is different, and that they came up with this final model where, depending on whether the membrane fluidity is increased or decreased, we have a higher affinity or lower affinity, or even no affinity at all of the ligand to the GPCR. I also would like to introduce a new member of the monolith family, which is monolith X, which was actually only launched last June. Why another monolith, or why would I talk about a monolith and now have a monolith X? So basically, the reason behind this is that we developed another technology, and additionally to the MST technology that I just explained, we have the spectral shift technology in this instrument, which is an orthogonal method to analyze interactions. And I quickly want to tell you how this works and also give you some examples for this. So when we're looking at a fluorescently labeled target molecule, we will have a distinct emission spectrum. And once this target is bound to a ligand, this will lead to either a blue or a red shift of this emission spectrum. These shifts can be really small, but we wanted to actually use this nature of ligand binding induced fluorescence change to analyze the binding. So what we do is we collect the fluorescence signal at 650 nanometer and 670 nanometer for a few seconds. And these wavelengths are just around the peak here. So we basically can look at the shift here. And we then plot the ratio of these two wavelengths over ligand concentration. And from this, we can then derive a KD. So for this, I also want to just show you a few application examples. So one quite interesting one is the 3C-like cysteine protease from the coronavirus, which is of course a potential drug target. And you probably know the nirmatrelvir as a component of the Paxlovid that was developed from Pfizer, and it binds covalently to the target. And we could, with very good signal to noise, determine the binding of this component to the protease. Another example that I would like to show you is the interaction between tau and the tau-5 antibody. So as you know, tau is found in neurofibrillary tangles in Alzheimer's disease, and tau-5 is an antibody that binds several tau isoforms and fragments, and that's what you can see here. So we analyze different tau isoforms to better understand what's the epitope that this antibody binds, and you can see that there are two different isoforms that are bound by the antibody, and there are certain fragments of these isoforms which are not bound anymore. So here the epitope is not present. And this is the last example. So here we're looking at a ternary binding and cooperativity. So this is the interaction between the PROTAC LC2 and the E3 ligase complex, as well as the PROTAC target KRAS. So for those who don't know, PROTACs basically bring the targets, so in this case KRAS, in proximity to the E3 ligase, which then attaches ubiquitin to the target, which leads to degradation of KRAS in this case. So to analyze these interactions, you would first make a control experiment where you just measure the interaction of KRAS to the E3 ligase alone, which is there's no affinity there. And then you would look at the interaction between the PROTAC and KRAS alone, and then, the PROTAC and the E3 ligase alone, and then add the third component, and you can see an increase in affinity as soon as you add all three components, which means that we actually have cooperativity here. Last, I would just like to highlight how what you need to perform measurements. So the instrument works with glass capillaries, and each capillary holds minimum amount of 4 microliter. So each titration point would go into one capillary, and then these capillaries are attached on a thermal element and then inserted into the instrument and measured. And there's the option of having single capillaries or a capillary chip for this instrument. And then we also offer labeling kits. So I actually talk quite a lot about which part has to be labeled and what kind of label was used. So of course, you can use targets that are already labeled, but we developed some fluorescent labels which are very sensitive to environmental changes, so very suitable for the methods that I presented, and therefore give you a better signal to noise. To conclude, I just want to highlight what you can actually do with the monolith. So you can measure a broad range of affinities from picomolar to millimolar range. You can measure many sample types, and you can also measure a broad range of interactions, so including protein-ion and protein-carbohydrate interactions. You can measure this KD independent of size and mass, and the measurement is in solution as it's in these glass capillaries, so you don't have to immobilize or anything. Also, it's basically very suitable with every butterfur that keeps your protein stable, so there are no really limitations on what you can add here. And also nice to have is that you can basically work without any maintenance and repairs, just because after each experiment, you would throw away the glass capillaries and it's ready for the next experiment. And we have a very user-friendly software with this instrument. So basically also people with no experience can walk up to this machine and use it straight away. And with this, I'm at the end, and I want to thank you for your attention, and I'm happy to take questions.
Thank you, Hannah, for a wonderful talk.
Thank you.
Does anyone has any question? I have a question. Does this technology exist in Ireland?
Yes, there are some instruments in Dublin and Galway. If you want, I can also share the people who have the instruments. So for example, Andrew Kellett's group at DCU has one instrument, and they use it a lot. It was installed this year. So yeah, definitely possible to contact them.
So, you know, in one of your final slides, it says that, you know, you can measure protein ion and protein carbohydrate interactions. I was just wondering, are there any kind of limitations there? There are certain ions that might not be able to be kind of characterized, or, you know, maybe certain sugars or polysaccharide or something like that, or is it, you know, pretty much anything goes?
Yeah, you can. So the general answer is anything works. Of course, if something has fluorescence at the same channel as you want to label your protein with, then that can be difficult, of course. But other than that, as soon as you can load your sample into a capillary, it's good to measure. And then, so of course, always, so the labeling step is really important. So that it basically picks up, you know, something binding. And there's sometimes you need to like try 2 or 3 different labeling strategies. So this is the optimizing part. But then as soon as you have the buffer conditions and the labeling ready, you can basically measure anything.
It's very cool.
I have a
Is
question.
someone?
Oh, yeah, sure, go ahead.
Okay, there is a question in the chat, but it is similar to the question I have. Maximilian is asking if it is possible to measure lipid binding to proteins. I also, my question was similar. It is possible to work in a liposome or nanodisc environment?
Yes, yes, you can definitely work with this. Again, you have to play around maybe a bit with the labeling. So one example I showed, they actually just labeled the nanodisc itself. You can also, I think one of my examples was the GFP fusion of the membrane protein, but it's no problem at all to go for these kind of things because it's in solution. You don't have to mobilize or anything. You have to think about the label. We do have different labeling strategies, which we offer. Of course, there are many out there. Fluorescently labeling is nothing new. So we, for example, offer histag labeling, lysine labeling, cysteine labeling, maleimide labeling. So yeah, there are options out there. And then of course for a membrane protein, it's more tricky because you don't have, you know, such a huge surface, but there are many examples of these things. Yes, thanks for the nice question.
What are the advantages over techniques like SPR or ITC?
Yeah, so for ITC, of course, you would use quite a lot of sample volume. And here, as I said, you have a minimum of 4 microliter per capillary. So if you think about the whole experiment, if you do a 16-point dilution series or something, you really have very small volume. And then for SPR, the main advantage would be that you don't have to immobilize. You can directly measure in solution, which can be tricky. Or if you think about the surface SPR, where you have to, just immobilize the target, there can be secondary effects, right? That something would aggregate or so just because of the surface that you have present.
I have a question. So how crucial is working with premium capillaries over the standard ones?
It really depends. So they are specially coated. So if you have something that's really prone to aggregation, you would like to use these, but very often also the standard ones work fine.
Any other question? So thank you, Hannah, for your presentation. And we are looking forward to the new Monolith X instrument. Thanks everyone for joining us today. We really appreciate you being here. If you have any further questions, please shoot an email to Dr. Hannah. We just put her email in the chat box. As you guys know, this webinar has been recorded, so we'll be very happy to share it with you later on. Thanks everyone, and see you soon.
Thank
Thank
you.
you very much.
Bye.
Thank you so much, guys.
Have a great day. Bye.